Your mission, if you choose to accept it, is to create a vaccine for the SARS-CoV-2 virus, or a treatment for the disease it causes, COVID-19. This mission is extremely difficult, and only the brightest minds will be able to accomplish it. The enemy agent, SARS-CoV-2, has infected over 6 million people in the US and killed close to 200,000 people. The virus spreads rapidly in dense households or workplaces, leading to a predominance of cases in people from ethnic and racial minorities, as well as nursing homes or high-density workplaces. If you are successful, you will stop many more deaths. The agent spreads through respiratory secretions from host to host and leads primarily to a respiratory illness. Most people survive, but many get severely ill and ultimately die. So this is your goal: create a treatment to save those infected, or create a vaccine to prevent infections. These are the different strategies you can undertake. Your choice. Block the virus with an antiviral. Help the immune system by giving prefabricated antibody like those in convalescent plasma. Regulate the host by blocking the cytokine storm with an immune modulator. Stimulate the host to produce immunity before the agent attacks, aka a vaccine. This is your target. Study it well. You need to know its structure and mechanisms if you are to be successful. These structures on the virus may be some of your targets: the envelope protein, the membrane protein, or the spike protein, RNA, or the lipid membrane. You need to learn how the virus invades, makes protein, and replicates. Blocking the virus at any of these steps is the key to stopping it. The virus enters the cell through special receptors by using its spike protein. Once in, it releases its viral genetic material called RNA. The RNA uses the host cell machinery to produce proteins that will keep the immune system at bay and help assemble new copies of the virus. New copies of the virus are assembled and carried to the outer edges of the cell. Each infected cell can release millions of copies of the virus before the cell finally breaks down and dies. The virus may infect nearby cells or end up in droplets that escape the lungs and infect other people. In most COVID-19 infections, the immune system responds appropriately and limits the virus, causing mild disease in the host. In severe cases, the immune system can overreact, overproducing molecules called cytokines that produce a dysregulated immune response called the cytokine storm, and this ultimately does more harm than good. Many molecules have or are being tested in this pathway, but to date, only 2 medications have high quality evidence that they improve patients' lives: an antiviral called remdesivir and dexamethasone, a steroid that limits the immune response. Vaccines are another option on your mission and may be the ultimate vehicle to eliminate the virus. Traditionally, vaccines have been made of dead or weakened organisms, but many novel mechanisms are being postulated for a SARS-CoV-2 vaccine. And those include vaccines based on proteins, vaccines based on genetic material like messenger RNA, and vaccines that use a virus to deliver coronavirus genes into cells. The ideal vaccine should be able to stimulate the host's immune system to produce memory immune cells that produce antibodies. The hope is for long-lasting immunity where antibodies or other immune cells protect the host on future exposures. Even if you find a molecule or vaccine that may work, the pathway to FDA approval is hard. Due to the pandemic, the timeline has been greatly accelerated, but still, studies need rigor and time to show that molecules work and are safe. Molecules go through 3 phases of clinical trials. First, small studies to prove safety, and then larger studies to prove that they work and still are safe. The timeline normally takes several years, but now you're expected to go through development in months. As of the making of this report, there are 315 treatments and 210 vaccines in development. You will have to build teams, as nothing can be done in a silo on your own. You'll not be able to do this alone. Good luck and Godspeed to you, sir or madam. This tape will self-destruct in 5 seconds. Hi, I'm David Goodman. I'm an infectious disease doctor at UCLA. As of taping this presentation, I'm actually the COVID-19 consultant at our main hospital. I tried to make this video lighthearted. However, you already know that this is a really serious disease and all of our lives have been affected in some way or another. I really appreciate your efforts in devoting your time to create content to educate around COVID-19, as we well have learned that misinformation kills. I'd really like to thank Rick Thomas and the National Academy of Sciences for the invitation to produce this video, and to my chief at UCLA, Judith Currier, for allowing me to participate. Please feel free to reach out if you need any help on any of your projects. And again, thank you for your efforts.